we've been studying risk factors for mental disorders for, well, over 2 decades now. And I always like to start with this slide because it kind of gives a general conceptual perspective on what we're doing. And I basically am calling this the envirome in neuropsychiatric development. So one can think about this as in terms of what we call life course perspective. It's a big issue in epidemiology, and the idea is to think about an initial insult that occurs in utero that may be any number of risk factors like infection, malnutrition, hypoxia, toxins, and I'll be mentioning some of these today. And so the idea of this model is that there's an initial insult that occurs in utero, and then through either physiologic or possibly epigenetic or other effectors, leads to a variety of abnormalities which manifest differently at different stages of development. And so at the time of birth, we see things like low birth weight, intrauterine growth retardation, and other findings like labor and delivery complications. And the idea is that these are then later risk factors for disorders that emerge later in the life course. So in childhood, of course, we see onset of autism and ADHD, delayed milestones, neuromotor dysfunction, social cognitive disturbances, and other problems. In adolescence, we see things like depression, anxiety disorders emerge, as well as the prodromal symptoms and decline in neurocognitive function that are characteristic especially of these adult onset disorders like schizophrenia and bipolar disorder. And the model also presupposes that these in utero factors interact with susceptibility genes and other environmental exposures, for example, and other triggers such as cannabis and stress for certain, some of these outcomes, not for all. So, and underneath each of these, underlying each of these are a variety of neural processes that many of you here are more expert than I am who do a lot of preclinical work. So this is just a kind of a general overview on the slide. So there have been a variety of developmental precursors of neuropsychiatric disorders that have been discovered in epidemiologic studies, and they include prenatal environmental exposures such as infection, inflammation, autoantibodies, toxins, hormones, and a variety of others. Perinatal events have been associated with a number of these outcomes, demographic factors like maternal age, anthropometric abnormalities, and I'll be talking a little bit later about head circumference, intellectual and cognitive development occurring premorbidly, familial genetic vulnerability, and the disorders that have been probably most studied with regard to these epidemiologic studies of neuropsychiatric disorders are autism, schizophrenia, bipolar, ADHD, but a variety of others that our group is also exploring in a birth cohort that I'll be telling you about. For people a little less familiar with the birth cohort study, and many of you are, so our ultimate goal is to identify early developmental risk factors for these outcomes. And in my view, a birth cohort study is the most methodologically rigorous and informative approach to address that question because you're studying a sample of people that you follow very early and collect prospective measures and then follow them over the life course for these psychiatric outcomes and intermediate outcomes. A birth cohort is a group of individuals who are born within a defined time period in a defined place and followed up for the outcomes of interest. And so when I got into this field, there were some studies, for example, showing associations of epidemics of influenza and schizophrenia among those who were in utero during those epidemics. And so a lot of the studies were really landmark findings because they were the first to report those associations, but they suffered from a variety of methodological limitations. And so we sought to improve upon the methodology, first by in 2 key ways, and there were other things that we did as well. But first is to have prospective ascertainment of exposures during pregnancy. And we did that through either prenatal biomarkers from archived biological specimens in individual pregnancies and previous systematically collected obstetric and perinatal records from centralized computerized registries. A second key issue in epidemiology and really in any clinical study is bias. And bias can lead to a lot of problems, spurious associations or the inability to find an association. So we use what's called a nested case control design, and in which a study is conducted among cases and a sample of controls from the cohort that gives rise to those cases. And the idea is if your cases and controls come from different places, that can bias your findings. So that's really a central design issue in all the studies I'll be telling you about. We also have, to the best we can, continuous follow-up, which allows, we use analytic methods that allow one to adjust for the problems of loss to follow-up, which can also cause bias. So the first study I'll be telling you about was in a place not far from here, birth cohort investigation called the Child Health and Development Study. This is kind of a preview to what I'll be telling you, and this we started this study in the 90s. And this was an investigation of all mothers and offspring who were enrolled in the Kaiser Health Plan in Oakland. I don't need to tell you more about Kaiser here, you all know it. And there were about 19,000 live births in this study. It was started in the 1950s and 60s. These are births from that period. And what they had available was really tremendous. It started not long after the thalidomide epidemic, so there was understood that there was a lack of knowledge about how medications and other factors in pregnancy altered later development. So they had a comprehensive database on prenatal, perinatal, neonatal exposures, parental background. But in addition, they'd collected blood specimens and took the serum from those specimens from all the mothers in pregnancy from different trimesters, and they stored them. And they're still there. And so when we did our study in the 90s, we were the first to look at psychiatric outcomes, including schizophrenia, and then later bipolar disorder. So we had about 71 cases of schizophrenia, we had 94 of bipolar disorder, but this study occurred much later than that one. And we followed them up using structured research interviews. So we used Kaiser electronic registries to identify people who were at risk, who we thought were likely, and then we confirmed them with structured interviews. We used one called the DIGS, which is modeled after the SCID, just for people who are interested in diagnosis. So I'm going to talk just a little bit about our findings. And I guess what we're most known for in that study was prenatal infection. And so in this study, we measured antibodies to influenza, and we validated it against seroconversion to influenza in the sample. And because we didn't have sufficient numbers of mothers, pregnant mothers, who had serum drawn at multiple time points. And what we found was that for the first half of pregnancy, the prevalence of exposure to influenza was significantly higher in the cases compared to the controls. In contrast, for the second half of pregnancy, we didn't see a difference. So this was the first serologically based evidence, I guess to my knowledge, still the only evidence that prenatal influenza may be related to the later risk of development of schizophrenia, and that was again in this cohort study. But we had about threefold increase in risk. So, and we next look at toxoplasmosis, which is an intracellular parasite. It's actually quite common in the population, and when an adult gets it or a child gets it, it rarely causes any problems. But if a pregnant mother gets it, it's been known to cause a variety of congenital abnormalities and severe sequelae, other severe sequelae. And what we showed here was we looked at those with moderate titers, so we measured toxoplasmosis titers, those in the moderate range, those in the high range. And for those in the high titer range, we found the cases were markedly overrepresented compared to the controls. So we had an association then between both influenza and toxoplasmosis and schizophrenia, and using biobank serum. So we looked at other risk factors in this sample, other infections, and then we calculated both odds ratios. We had the odds ratios, but then we looked at the population attributable fraction, which is explained here. It's the reduction in the incidence of disease that would be achieved if the population had been entirely unexposed compared with its current actual exposure pattern. So it's difficult to say that an infection causes this percent of the disease. It doesn't quite work well when you do that. There are a variety of reasons. But what you can say is, if you were to eliminate all cases of the infection, which of course is not feasible, but if you were able to do that, how much of a reduction in risk would you see? So, and we calculated that, I don't have error bars here, but we calculated that for influenza, about 14% could be eliminated, toxoplasmosis, 13%. So if you add all these up, 6% here, granted, we can't eliminate a third of all cases by getting rid of these three infections, but it does suggest that we perhaps, by preventive measures, assuming a causal relationship, we could reduce the risk incidence of schizophrenia in the population by, fairly substantive amounts. And certainly these factors are higher than most of the susceptibility genes that have been looked at in schizophrenia. So it tells us basically that environment is important. That's the main message of this slide. So we then moved on to the bipolar disorder study, and the idea was that this Kraepelin himself had said, well, there was this kind of period, this overlap between, this is schizophrenia and this is bipolar disorder, and there's this kind of overlap between the two of them in terms of their clinical presentation, and he looked at other things as well. He did a lot of biological research as well. So it led us to then pursue a study on bipolar disorder, and we looked at the same risk factor here, which is maternal influenza. So we first looked at clinically diagnosed influenza using the Kaiser records, and we found a nearly 6-fold increase in the risk of bipolar disorder with exposure to influenza in pregnancy. And then we looked at the sera. So these are just done separately. We looked at influenza antibody here, and we used the exact same method that we did in the schizophrenia study, and we found almost the same effect, about a 5-fold increase in risk. So it seemed to be, from these findings, that perhaps influenza could be not only a risk factor for schizophrenia, but also for bipolar disorder, and that raises a number of questions about pathogenesis and specificity that we can talk more about. So the issue, as I showed you those sample sizes, we had 70 cases of schizophrenia, we had 94 of bipolar disorder. That was fine, and it led to these findings. But the biggest issue that we had really was it's just not enough subjects to really get first a robust enough findings, and I think it also kept us from looking at a variety of additional questions, like what are effects by gender, and if you stratify on other factors. So about 10 years ago, I found that there was this cohort called the Finnish maternity cohort, and it wasn't well known. There were only maybe 4 or 5 papers published on this cohort, and no one used a national sample, and no one had looked at psychiatric outcomes. And what I really wanted, though, was to do the same thing that we did in California, in the Kaiser cohort, do almost the same kinds of things, where we had prospectively collected serum samples, we measured a variety of biomarkers in the sera, and then looked at relationships with later outcomes among those, from the pregnancies, the offspring of the pregnancies, to see if there were increased risks with certain risk factors that we tested for. So the Finnish maternity cohort contained virtually all pregnant women in Finland from 1983 to the present. About 1.5 million pregnancies. So mothers in Finland are required to be tested for hepatitis and HIV and rubella and syphilis. And that's true in a lot of places. But in Finland, what they did that was quite unique is they took the remaining serum and they put them all in one biobank, in one freezer, because all the serum got tested at the same place, which is in Oulu, Finland, which is in the northern part of Finland. And so we had, and the population is very compliant. I don't know if we could quite do something like this in the US, maybe not. But they say get yours tested, they get tested, and only about 1% declined. And so they had this huge freezer where all the serum were stored. And these were archived, and most of the pregnancies were quite early, months 2 to 4 of pregnancy. And I think that has implications for some of the hypotheses that we were testing, which is we thought that a lot of these exposures in early and mid-pregnancy, especially infections, might be the most important period. So like a lot of Scandinavian countries, and I was influenced to some degree by the work that had been done in Denmark and also in Sweden, where they use these national registries, because they all have socialized health care. They have real universal health care there. We're working towards that, but it hasn't fully worked yet, but we're getting there. And so they have hospital and the government pays for all their health care. So they have hospital and outpatient discharge registries, so they have all recorded diagnoses for psychiatric hospital admissions and outpatient visits. And there are other registries that we were able to link all together. So we linked the Finnish maternity cohort, that is the mothers of those offspring that we wanted to follow, with the offspring themselves throughout the course of their medical history, their diagnoses. We also linked them to medical birth registries. So we looked at a variety of pre- and perinatal conditions. We also had longitudinal developmental data, population registry, which allowed for a collection of additional covariates, but in particular, we could link family members too. So we could link whole families together. And we had they use these PIN codes, like a social security number that allows for the linkages. And this very briefly shows just the schematic of the methodology that we used and what we call the Finnish prenatal studies, or the other term I use is the FiPS. So we had, it turned out to be about 2 million births once we got to the end of the period. The autism cases were born from 87, 2005. Schizophrenia was a little bit earlier. And so the key here is we had the case identification, and this was the period of follow-up. And we had the identification of the controls matched on sex and date of birth from the Finnish medical birth register in a 4-to-1 ratio. And from that, we generated a case and control database. And we then also we collected their serum specimens from the freezer. We also collected, it wasn't computerized, so all the data that I've told you about is computerized registry data, which makes linkage a lot easier. But this was a lot harder to do because we were very interested in developmental data, like head circumference over the course of treatment from well baby and child health clinics, and developmental milestones. And they did a spectacular job in Finland going to the clinics, actually throughout Finland, and collecting the data and abstracting the records. And I'll be showing you some data from that later in the talk. The medical birth register I mentioned, and the population register. So the key is all these registers were linked together, and that was really important for being able to do this whole study. So the design advantages, well, first, the country is the cohort, and we had nearly complete ascertainment of all diagnosed schizophrenia, autism, and bipolar disorder cases in Finland. We were able to ascertain biomarkers of these exposures then with the sera, as I mentioned. And the key was we got quite large samples. So at present, we have about 4,000 cases of the ASD, the broad ASD. This I'm going to refer to later, childhood autism, the narrowly formed autism. They call that in the ICD system childhood autism. And I'm going to go back and forth a little between that and ASD for reasons I'll explain. Well, I'll tell you now. So the child autism cases really were what we included in a lot of our serologic studies. Part of the reason was cost, but part of the reason was also that we've had validated childhood autism diagnoses using the ADIR, and we had not done validation work on the other autism subtypes. But there are other analyses I'll be showing you where we look at broad ASD for analyses that require the larger sample sizes. And those involve registries rather than the prenatal serum specimens. We have what's called a FiPSA then for autism, FiPS for schizophrenia, about 1,500 cases, and bipolar disorder, a good number. And we have also, I'm not going to discuss this, but we've published also on ADHD, and we're currently pursuing that. So, what it allows us to do with these large samples, a variety of things, but one is to look at the interactive and mediating effects between different prenatal exposures and perinatal complications. Now, because we had uniformly collected subjects, samples, same country, same biobank, same registries, we could look at diagnostic specificity, I think, in a more robust methodologic way than if you were to say, look at different samples in different countries and different disorders. So that created some uniformity here, and I'll be talking a bit about that. I'll be showing you some data where we looked at both autism and schizophrenia. We were able to minimize selection bias because the key was that we had nearly all the pregnancies in the country and nearly all the cases of these disorders in Finland and representative matched controls. So if you have nearly all the cases and nearly all the diagnoses, it's very hard to lose people selectively who have a certain diagnosis combined with a certain exposure. And that can cause significant problems with bias. And then we were able to leverage the power of developmental trajectory data to address intermediate phenotypes over different periods during the life course. And I'll be showing you an example of growth velocities, infant growth velocities of head circumference in autism. So we started, looking at just some of the basic, risk factors that have already been identified. And at the time, not many had been when we first studied these, but we started, people here have found these associations as well in cohorts that they study. So we found that hypertension was associated with risk of autism, also APGAR score, less than 7, and monitoring in the NICU, so neonatal treatment. And we also looked at, here now we're looking at a couple of different ASD subtypes, childhood autism and Asperger syndrome. And this is just the way the study evolved. Here we looked at certain subtypes. And what we found here was, as has been shown, very low birth weight being associated with about a threefold increase in risk of autism, childhood autism, and then just low birth weight, but not very low, also associated. Preterm birth associated, and wait for gestational age. So this kind of, in a way, validates the quality of the registers and the fact that we had done the study, we think properly, and also these served as potential covariates to be looked at in our studies, in the studies I'll be telling you about. Interpregnancy interval, I think, is something that my colleague, Kili Cheslak-Postava, found in a California birth cohort, and she started working with me, did a fellowship with me, and started working with me, and wanted to study that in Finland, and said, why not? So it's a potentially modifiable factor influencing prenatal environment. The short interpregnancy interval could be an indicator of the depletion of maternal nutrient reserves, like folate, which Rebecca Schmidt studies, or unresolved inflammation from a preceding pregnancy. And it's also associated with risk factors like Short-Walker studies, preeclampsia, and unintended pregnancy, and a higher risk of maternal risk behavior. So it's a kind of a marker of a variety of exposures that could potentially precipitate risk of autism. And so what she found in Finland then was that a shorter interpregnancy interval, she replicated her previous finding from California, about a 1.5-fold increase in odds for those with spacing of pregnancies less than 12 months. And then interestingly, and what she didn't find in the other study, was that long interpregnancy interval was also associated with risk of autism. We don't have a good explanation for the later findings, so I'd be interested to hear, and if you have any comments about that in our study. So it seemed to be kind of a U-shaped distribution. So moving on to infection, many of you, we've been talking all day about the maternal immune activation model. Many of you are studying that, and that was clearly a big interest of mine from our earlier work. And so we still to date can't measure cytokines, inflammatory biomarkers in these particular class of inflammatory biomarkers in these samples, but we could look at C-reactive protein. CRP is an acute phase reactant. It's an established marker of low-grade inflammation from both infectious and non-infectious exposures, and it's substantially elevated during acute infection. And so it can be induced not only by infection, though, but also by chronic inflammation, tissue injury, and other factors, which then leads to the production of pro-inflammatory cytokines, which are then transduced through the liver, and which produces the C-reactive protein. So we measured this in 677 matched case-control pairs in Finland, in the maternal sera in Finland, and we found that for the greater than 80th percentile of the distribution of C-reactive protein, an odds ratio of autism of about 1.4. That's adjusted for many factors. And these lower levels, we didn't see an effect. We also saw an effect using log transform, which I'll show you in 1 minute, looking at CRP as a continuous exposure. And that's here. So for autism, for every 1 unit increase in CRP, we saw a 1.14-fold effect. And then interestingly, we also looked at schizophrenia, and we had an even bigger effect. So, and then we also looked at CRP levels greater than 10, and that's been thought about as being a particularly good marker of an acute insult, such as an infection. And we had a pretty good sized effect for autism, and also, again, for schizophrenia. And this brings up this question about specificity, that the risk factors that we're studying may be risk factors for more than just one disorder, and it was nice to see this given our previous findings in the Kaiser cohort, and now we've shown it for autism. So this is the longest information slide I'll show, but I really wanted to do justice to this, and I'm not going to go through it in great detail, but this is sort of what's been studied and known, and it doesn't include everything, but about maternal and neonatal infections and inflammation in ASD. So in a study, from Zerbo, it was actually lower maternal CRP levels were related to ASD. And one thing in preparing this talk, though, is I realized that they were looking here at a little bit different time period, a little bit later in pregnancy, months 15 to 19. As I mentioned, we were looking at a fair amount earlier in pregnancy. Could that have given rise to the difference? We don't know, but it's certainly worth noting that these findings obviously are the opposite. There's the EMA study, which showed maternal cytokines being elevated IL-4, IL-5, interferon gamma, and also in the EMA study, a variety of other cytokines have been associated with risk of autism, as well as certain cytokines, again, associated with decreased risk of ASD. So it's a complicated story. I'm not here to say it's very clear there's elevated cytokines in mothers of autism, and certainly mothers of cases of autism. It's a complicated story that we need to work through, and some of these cytokines related to a decreased risk are also somewhat pro-inflammatory. There have also been studies looking at, this is looking at serum or blood. This is a birth cohort study in Denmark that looked at amniotic fluid and also found elevations so with some overlap between some of the earlier studies in cytokines. Regarding infections, specific infections, the Danish cohorts, the CHAP study and Kaiser study from Lisa Krohn, the viral infections in certain trimesters and bacterial infections have also been related to autism risk, and we're currently working to look at specific agents, most notably influenza. We have influenza antibodies measured in our autism sample. We have not yet analyzed those data. And of course, then there's the preclinical work, much of which is done here, Melissa Baumann, on showing that in rhesus monkeys, a variety of phenotypes that have been observed in autism have been associated with the maternal immune activation model, for example, using poly-IC. And now we go to autoimmune disorders. So there had been a good previous literature on a variety of immune conditions that have been associated in probands and in family members with ASD. And they're listed here, like rheumatoid arthritis and psoriasis, et cetera. And this then goes to Judy Vanderwater's work and many others here who have been looking at brain autoantibodies. This really fascinates me. The idea that a brain autoantibody, IgG reactivity against fetal brain proteins, can lead to, during pregnancy, can lead to an increased risk of autism in the offspring. So, and these are some of the findings that have been observed, and I'm sort of presenting to you what a lot of you already know, but the anti-fetal brain antibodies in the mother being related to lower expressive language in the child, abnormal brain enlargement, and then a variety of these autoantibody or antibodies to these markers, which Judy has been studying extensively, found in a paper published in 2013, 7% of mothers of children with ASD, but very rare in controls. And I find this to be very convincing evidence of a role of autoantibodies in autism. In this study here by Brimberg, that children with ASD preferentially carry antibodies to fetal brain tissue. So we first studied this in our cohort just by looking at clinical diagnosis of autoimmune diseases. So we had both probands and we had mothers, fathers, and siblings. And we found overall an increase in risk, or an increase in risk of autism in probands who had autoimmune diseases. In general, this is sort of combining them all together. And then we looked at the mothers, fathers, and the SIBs, and we found actually the strongest findings for the moms and the SIBs, and a finding for the fathers that just narrowly missed significance. I thought this was interesting, though, given the maternal finding, given the effects on the in utero environment, and siblings likely share in a utero environment. I found that to be interesting, but it's difficult to say that those findings are really different. But again, what it tells us is that in this large birth cohort, where we had 4,600 cases, that we see these associations of familial and proband autoimmune disorders and autism. We then looked by body system. It's a sort of a different way to look at it because we didn't have a big sample, but we wanted to group them in some way that made sense. And what we found, interestingly, though, was for the central peripheral nervous system, about a nearly 5-fold increase in odds if they had an autoimmune disorder that affected that system, odds of autism. And also, autoimmune disorders that affect the endocrine, the gastrointestinal, the respiratory system, skin and mucous membranes. So it seems to be this kind of general effect of different autoimmune disorders affecting different systems that can be related to an increase in risk of autism. But I was really interested in kind of looking at a specific autoantibody too, just like Judy has been studying. And so we did a study where we looked at thyroid autoantibodies and thyroid hormones and the mother's serum, which we quantified. And to our knowledge, we didn't know of a maternal autoantibody related to a specific autoimmune condition that had been examined in relation to autism in the offspring. So we looked at this autoimmune thyroid disorder. Mostly it's Hashimoto's thyroiditis, although the marker that we looked at can be a marker of other autoimmune thyroid problems. And we measured what's called thyroid peroxidase antibody, an antibody involved in autoimmune thyroiditis in the maternal sera during pregnancies of the autism cases and controls. And here we showed that higher prevalence of the TPO antibody in the cases compared to the controls, with an odds ratio nearly twofold. And then we looked by sex and by comorbid intellectual disability. So these are all the subjects. And then we've had the finding stronger in males than in females. But we had some finding in females, and it may just be due to the lower power because of lower sample sizes. The finding, interestingly, was related to those that didn't have intellectual disability, not those that did. So it's hard to argue it was related to intellectual disability. And then we looked at maternal thyroid disorders and childhood autism. So now we looked at the hormone itself. And the question was, is it that the hormone caused the problem, or was it the antibody? Interestingly, none of the clinical conditions of hypothyroidism or hyperthyroidism were related to autism. So it tells us that it wasn't the hormone, it's the antibody, which fits more with the autoantibody hypothesis. We also looked at the hormone itself, the levels of the hormone, like free T4, that may not have even been subclinical, just to see if there was a relation of hormone level, and there was no association. So now I'll move on to persistent organic pollutants. These are lipophilic, halogenated organic compounds, and we focused on two main types. One was DDT, and that's best measured by the metabolite PPDDE. And we also looked at polychlorinated biphenyls in the maternal sera. And these were in widespread use in developed countries. DDT as an insecticide, PCBs and transformers and electrical equipment. Now they're more in the developing world. And they've been banned in the developed world for many years, but they remain in the environment. And that includes the United States, it includes Finland. They have a half-life up to several decades, and there's continued exposure of human populations through a variety of means, sources. Contaminated waste sites, soil and plants, PCBs, consumption of particularly large fish, and contaminated water. All of us have these in our system, and the mothers in Finland had them in their systems while they were pregnant. And there's ongoing prenatal exposure. These environmental toxins are transferred across the placenta, and the cord blood concentrations are 1.3 times as high as in maternal plasma. So they're actually more concentrated in the fetus than they are in the mother. PCBs have been associated with a variety of neurodevelopmental outcomes, like motor behavior, novelty preference, lower cognitive processing, lower full-scale IQ. Behaviorally, they're associated with ADHD-related behaviors. And as most of you, all of you know, neurocognitive deficits and ADHD commonly occur in ASD. And also maternal DDE has been related to prenatal and perinatal complications associated with ASD, including preterm birth and low birth weight. So there have been a variety of prior studies. One most notable was by Lyle et al. From Lisa Crohn's group, showing increased maternal levels of several PCB congeners, and they're listed here, and that was the EMA study. There weren't other studies that looked at autism per se and maternal POPs within using biomarkers, but there have been ecologic studies, include those conducted here, for example, by IRVA, looking at looking at residential proximity to toxic waste sites, looking at DMTP and ASD and finding an association in girls. And Rebecca Schmidt showed that folic acid is related to likely an attenuated association between maternal pesticide exposure and ASD. So this suggests suggestive evidence. Again, it's an association, it's not causation, but it suggests that these maternal neurotoxins can affect fetal brain development, that we know affect fetal brain development, can be related to risk of autism. And this study, it's now in press in the American Journal of Psychiatry. And it's coming out next month. So our main hypothesis was that POP exposure, either DDE or PCBs, can be related to the risk of autism, and to see if these associations are modified by sex and intellectual disability. And this is our methodology that we used, gas chromatography, tandem mass spectrometry, standard methods. And we looked at a variety of congeners, DDE as one measure, and then the variety of congeners, which represent 80% of all PCBs on a mass basis. And we looked at the Pearson correlation coefficients and found, as one might expect, that DDE was not that strongly, was related to, but not that strongly related to PCBs. And the PCBs were obviously very strongly related to one another. So here we show the maternal levels of first DDE and then the total PCBs. And we found that the mean levels and the median levels were greater in the mothers of cases compared to the mothers of controls. We had nearly 800 matched case control pairs. Total PCBs were about the same in both the means and the medians between our cases and our controls. These here and these here. And I'm not going to go through this in detail, but we did look at individual PCB congeners because we wanted to see, could we get the same findings that Lisa had? And looking at those, the 138 and the 153 were also no different. So we actually looked really carefully at that, and we analyzed our data. Like, they analyzed their data a little different than we did. We did it the same way, and still there was no association. And this shows the odds ratios. So for PBDDE greater than the 75th percentile, we had an adjusted odds ratio of 1.32, which was statistically significant, but odds ratio of 0.95 indicating no association for PCB levels greater than the 75th percentile. So, and that's adjusting for several covariates, and we looked at other covariates for associations with the exposure and with the outcome, and tested them quite extensively. And this doesn't seem to be confounded by the covariates, at least that we had in our data set. And then we looked at by gender and also by the presence of comorbid intellectual disability. So we found the finding in the males, but not in the females. But we looked for the p-value for interaction, and this does look quite different, but the p-value for interaction was non-significant. For ASD plus intellectual disability, we had a greater than twofold increase in risk of following exposure to elevated levels of DDE. For PCBs, as we thought maybe we'd see some finding, either for males, for females, but again, pretty similar between the males and females, and for those with or without comorbid intellectual disability. So when we submitted the paper, it got rejected, the American Journal of Psychiatry, and they said, well, you're looking at too many things and things like that. And we said, well, we really had the two hypotheses, so they believed us on that, but then they said, well, why would it be that DDE, DDT, and PCBs would have different effects? So it took me a long time to think about this, and I think this is still just speculation, but I needed an answer because I wanted the paper to get into that journal. I just put a lot of work into it, and I thought it was worthy of American Journal. I don't know. So I said, so it turns out, actually, I looked at papers. There was a paper by Matt Longnecker, who's been studying these persistent organic pollutants for many, many years. And he had shown that maternal exposure to DDT and DDE were related to premature birth and small for gestational age status, both of which are well-replicated risk factors for autism. And I looked very carefully to see if PCBs were related to these, and it doesn't look like they are, at least from the studies that he's done. DDE also is known to inhibit brain androgen receptor binding, androgen-induced transcriptional activity, and androgen action, including in the developing rat brain. And interestingly, the offspring of rats infected with valproic acid, a known in utero risk factor for autism, exhibited reduced androgen receptor expression in the cerebellum. As many of you know, in study, cerebellar abnormalities have been demonstrated in the brains of cases of autism, as well as in rat offspring exposed to prenatal valproic acid. And in contrast, it appears to be that PCBs largely increase and not decrease brain androgen receptor expression. Now, I don't have a theory in terms of, you can think about androgens and gender differences, and I haven't taken it that far yet, but that seems to me at least to be, at least some it provides us with some biological explanation for why we saw those differences between DDE and PCBs. Now, as I said, we had the finding. The finding was mainly driven by individuals who had autism and comorbid intellectual disability, so it raises the question that maternal DDE is related to intellectual disability in general and not autism. However, there are many studies that have not demonstrated associations between DDE and intellectual disability in general. So it's conceivable that maternal DDE could also be a risk factor for a subgroup of autism cases who also have intellectual disability, but a limitation of our study is we don't have a group of intellectually disabled individuals who don't have autism, and we'd need to do that to really confirm it. So what we're doing now is looking at the specificity of maternal POPs to autism. And we now have a new grant funded by NIEHS to evaluate associations between maternal POPs and other diagnoses. In this case, we're looking at schizophrenia and attention deficit hyperactivity disorder. We're also evaluating additional environmental chemicals, or we aim to do this in our future work in the maternal sera. And we're trying to encourage people, including at Columbia, translational research to see if we can better understand, first confirm the plausibility, biological plausibility of this in utero risk factor, and potentially look at some causal mechanisms like those that I hypothesized when I was talking about the androgen receptors. So I'm going to shift gears now and talk about a postnatal risk factor, and that's childhood head circumference. I had a nice talk with Dave Amaral about that today. And so our findings don't fit with everybody else's findings. They do to some degree, but there's some overlap. So as a lot of you know, elevated head circumference has been found in individuals who were later diagnosed with autism, as well as macrocephaly, as well as elevated head circumference at birth and throughout infancy. But the periods differ between studies. There are also some negative findings. And previous studies had a number of, I think, really landmark studies that established this as a possible risk factor, but they didn't have direct measures of actual growth velocity. They did analyses that didn't look directly at the velocity of head growth. They generally had small sample sizes. They had head circumference data at relatively infrequent intervals over kind of long stretches of time. And some of the studies, actually, quite a few, used clinical and population reference norms rather than a matched control group. So in our study, we aim to examine the relationship of growth velocity of head circumference in the first 2 years of life using longitudinal data. And we use those data that I told you about earlier that we abstracted from the Well Baby clinics. And we had 468 matched autism case control pairs, a minimum of 6 longitudinal measurements per subject. And these are all, again, in the first years of life. So it's a little more densely spaced than in other studies. In addition, my colleague Ian McCaig, who's a professor of biostatistics at Columbia, had already developed this non-parametric Bayesian modeling approach that could quantify growth velocity specifically. We had childhood autism only, again, that narrowly defined subgroup, and as you saw, a fairly large sample size. So these are our findings. What you're seeing is the mean difference of head circumference growth velocity. So a number above, or you'll see points above this line, mean it's increased in the cases, and below it is decreased in the cases. But I wouldn't be showing this to you if it wasn't increased in the cases. So, and this is looking now from birth, age 6 months, 1 year, up to 2 years. And interesting, what we saw was that the risk was the highest, actually, the growth velocity and risk was the highest in these very early period of life. This is about months 2 to 4 of gestation. And previous studies hadn't shown it that early. Many of them didn't measure it that early. They had measured at birth, but not at these time points in the middle. And so you can see here they are elevated at birth. What this suggested, though, was that early head circumference growth velocity could be a risk factor for later development of autism. In this, at least in this Finnish sample. Then we did the exact same thing in schizophrenia. We got their records, we abstracted them the same way, analyzed their data the same way. And in females, we get a very, very similar pattern female offspring with schizophrenia. Very similar patterns to what we saw in the overall sample. Now, we did show it also, I'm not showing these data now, for both males and females in the autism sample. But in males, there was nothing. Again, we don't know what this means. We don't know why we're seeing that, but I think it was curious that we saw that similar pattern, that very early period of after birth being related, head circumference velocity being related to schizophrenia. So what are the explanations? I mean, they really go on, but, and here I give some possible explanations. Not all these findings have been replicated, but perhaps excess neurons, mini-column numbers, premature expansion of dendritic and axonal arbors. And in schizophrenia, it doesn't quite fit with what we understand in schizophrenia because it's actually more decreased head circumference at birth, but there are very few studies, and they're not national samples, and they're not all population-based. What might cause this? Again, a speculation, but something that could be followed up on are neurotrophins, like insulin-like growth factor, which modulates brain growth and myelination. And there's also higher plasma levels of insulin-like growth factor 1 is correlated with head circumference in autism. So the question that we're addressing now is, are deviations in head circumference an indicator of some prenatal exposure, an earlier exposure? And we have all the data, of course, on these same subjects on their prenatal experience, and we're working now to try and relate these prenatal biomarkers to head circumference growth velocity. So the FIPS cohort it's not a perfect cohort, so we don't have blood specimens, so we can't look at DNA. There are some analytes, as I mentioned, we have had difficulty looking at cytokines in the frozen stored sera, but there are many things we can measure. So for all the environmental toxins, we've gotten very high recovery rates. As I mentioned to you, antibodies can be well measured, CRP and hormones, and we've looked as well as I told you. So we have data on mental retard or intellectual disability, but not systematized data on IQ, which would be really nice to have, but we don't have that. And as many of you know, CRP levels and other inflammatory biomarkers are related to obesity. I had a good talk with Cheryl Walker today about that, but we didn't have body mass index in the cohort, so we can't control for that covariate. So we're moving on to one of the final slides now, which is what I call mining the envirome. And it's kind of grandiose, but it's the idea that first we can identify potentially new exposures. So we're currently funded now to expand the size of the cohort by about twofold, so as a number of cases by twofold, by longer follow-up, because I mentioned they were followed up to 2009. In our new grant, they'll be followed up till 2016. So we expect to get about double the number, maybe up to 8,000 cases of ASD and over 3,000 with schizophrenia. And we're also, as I mentioned, looking at diagnostic specificity to ASD by looking at schizophrenia, ADHD. We hope through our work, by looking at certain other biomarkers and animal studies, to look at both convergent and divergent pathophysiologic pathways that could explain why we're seeing associations between these prenatal biomarkers and some psychiatric outcomes. And in some, we see similar biomarkers for things like schizophrenia and autism. Why is that? These are all open questions, and we hope that can be investigated further. Gene-environment interplay, I think, would be important. We can't do that in this cohort. There are some existing cohorts where there's the potential to study G by E interaction or mediation. For example, C-reactive protein may be an indicator of a genetic polymorphism, and that was studied using Mendelian randomization, at least a Crohn's study. And then, as always, paying service, rightly so, to both the possibility that these exposures could cause epigenetic alterations. This clearly has potential implications for the prevention, as many prevention of these disorders, as many epidemiologic studies do. And I mentioned the possibility of translational studies through animal models, and including those being done here. So finally, I'd like to acknowledge my collaborators. This is a really big team effort, both in our Department of Psychiatry at Columbia and the Department of Biostatistics. The University of Turku in Finland played a central role in the collection and the analysis of the data. This is the place where we got the maternal serum specimens, and people also that helped us, especially with the analysis of persistent organic pollutants. So it's really a very big team effort. And I'd like to thank you all for your attention.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.